Good morning to you all present over here. Okay, last time we discussed chromosomal theory of inheritance. In that, we discussed 2 points. Okay, but we have to know that who has given this chromosomal theory of inheritance. That was given by Sutton and Boveri. So what he does, he correlates chromosome as well as Mendelian factors. Okay, and that Mendelian factors was replaced by gene, and who replaced this word? So that was replaced by Johansen. Mendel used that as a unit factors or Mendelian factors, but gene was given term by Johansen. Okay. So yesterday we seen 2 points. One points we seen that hereditary traits chromosome same retaining the number like Mendelian factors are retaining. Okay. Next also what we see that both chromosome and genes occurs in pair. Okay. Now today we'll discuss another point, chromosomal theory of inheritance. Okay, so this today we'll discuss that first two we seen that a chromosome containing same number like genes. Then what we seen both are having in pair. Then today it is what both chromosomes and genes. Okay, both chromosomes and genes, they are containing what? Gamete. So a gamete contains only one chromosome type. A gamete contains only one chromosome type. Okay, a gamete, because male gamete is there, female gamete is there, then fertilization will occur. So a gamete contains only one chromosome type. Okay, and only one of the two alleles are there. So and only one of the two alleles of a trait, and only one of the two alleles of a trait. So this was postulates of chromosomal theory of inheritance. Okay. Then what was that? This paired condition, both chromosome as well as Mendelian factor, is restored during fertilization because male gamete contain half, female gamete contain half. Okay. So when fertilization occurs, so what happens? They are restoring. Okay. So the paired condition of both chromosomes and Mendelian factors is restored. The paired conditions of chromosomes and Mendelian factors is restored during fertilization. Fertilization means this male and female gamete, what happens? They will come close to each other and form zygote. Okay, so during fertilization, what happens? These paired conditions of chromosomes as well as Mendelian factors, these both, okay, paired conditions of both chromosomes and Mendelian factors is restored during fertilization. Okay, now another thing is that Sutton and Boveri also find out that these homologous chromosomes, okay, these homologous chromosomes, synapse, they are mingling, okay, synapse during which cell division? That is occurring during meiosis. So, homologous chromosomes synapse during meiosis cell division. Okay, they are synapsing during meiosis, cell division, and then what happens? Then separate and segregate. Okay, and then what happens? Separate and segregate independently. So, and then separate or segregate independently. Okay, then they separate, then they separate or segregate what? Independently into different cell. Into different cell. Okay, they are separating or segregating into different cell. And what happens? Which is the basis of segregation and independent assortment? Okay, this is the basis for what? Segregation and independent assortment of hereditary traits. So which is the basis for segregation and independent assortment of hereditary traits. Okay, here you have to remember that always synapse occur, this homologous chromosome synapse occur during which cell division? That occurs during meiosis. Okay, and this was complete information known as chromosomal theory of inheritance. Okay, now what I told that Johansson given the term gene, many time I have seen, so you have to remember that one. Johansson coined the term gene for what? Mendelian factors. This you have to remember. Johansson coined the term gene for Mendelian factors, or we can say replace gene with Mendelian factors. Okay, so who coined the term gene? That scientist name is Johansson. Or who replaced gene with Mendelian factors? So that scientist name is Johansson. Okay, now we will see that what are the points that comes under, we seen that cell division. So how this chromosomal theory of inheritance impacts on cell division, that we have to see. So what we have to see? Points correlates with cell division, correlates with cell division. These are the information that already you took in class 11th. Okay, but I am just a quick reviewing because in depth we cannot go inside, but in quick review you have to remember that first is what? Cell division means DNA replication. So DNA replication occurs in S phase, synthetic phase of interphase. So first is what? DNA replication or duplication takes place at S phase of interphase. Okay, so DNA replication occurs or takes place at which stage? That is S phase of interphase. Okay, then what happens? Synapsis occurs. Okay, so in homologous chromosomes what happens? Synapsis occurs. Okay, each pair of synapsed chromosome, synapsis occurs. So each pair of synapsed chromosome is known as a bivalent or tetrad. Okay, so what we can say in meiosis, in meiosis, each pair of chromosome synapsed. Okay, so in meiosis, each pair of chromosome synapsed. And what synapsed? Homologous chromosome. So in meiosis, each pair of chromosome synapsed homologous chromosome and known as what? Bivalent. Okay, so this is known as bivalent. Okay, then what happens? These bivalent chromosomes align on equatorial plate, and that stage is known as metaphase. Okay, that stage is known as metaphase 1, especially because meiosis containing two steps. One is meiosis 1 and one is meiosis 2. So I'll not go inside that, but in quick review I'm just reviving your remembrance. Okay, so the bivalent chromosome align. Next, the bivalent chromosomes align at equatorial plate at metaphase 1. What is the stage where bivalent chromosomes align at equatorial plate? So that is metaphase 1. Okay, then what happens? Next is anaphase 1, where what happens? These homologous chromosomes separate, but sister chromatids remain attached. Okay, so now come on that. In anaphase 1, homologous chromosomes separate from each other. But their sister chromatids remain attached. Their sister chromatids remain attached to each other. Okay, so metaphase only homologous chromosome separated, but sister chromatids remain associated, remain attached. Okay, and this remain attached to each other with the help of centromere. Okay, then next stage is what? Anaphase 2. There what happens? This splitting of centromere occurs. So in anaphase 2, splitting of centromere occurs. Okay, so splitting of centromeres occurs at which stage? That is occurs in anaphase 2. Okay, and what happens? Movement of chromatids, then chromatids separated, so movement of chromatids towards opposite pole of the cell. And what happens? And movement of chromatids towards opposite poles of the cell. Okay, so this is what here splitting of centromere and chromatids movement. Okay, chromatids movement towards opposite poles of the cell, and that's why what happened, meiosis results 4 haploid daughter cell from one diploid parent cell. So what happens, meiosis results 4 haploid daughter cells from diploid parent cell. Okay, so meiosis results in what, 4 haploid daughter cells from a diploid, from one diploid parent cell, or from one diploid parent cell. So these all coming under what, chromosomal theory of inheritance, that is correlates. Now how it is happening, that we will see with the example of image or figure. Okay, so we'll see that, then everything will be clear. Now in, we already know that interphase containing many stages, G1 is there, G2 is there, S phase is there, M phase is there. Okay, but here we will show in a G1. Okay, so in G1 what is that, we'll see that in G1 stage. Okay, so in a G1 stage what is that, one is from male and one is from female. One is from male and one is from female. Okay, so one gamete came from male and one gamete came from female. So that is under G1 phase. Okay, then what happens in G2 phase? This, what happens? Chromosomal duplication occurs here. So in G2 stage. So here what happens? Duplication occurs, okay, and they contained in a central formation, that is a centromere. Okay, now then what happens in meiosis anaphase? Bivalent was there. Okay, so what happens? Sister chromatids remain attached but homologous chromosome separated. So come on, meiosis, meiosis. In meiosis, it is anaphase 1. Okay, so here what I told, that So here what happens? Centromeres remain attached, okay, but their homologous chromosome separated. This centromere attached, but homologous chromosome separated, okay. Then what happens in meiosis 2? This is meiosis 1, anaphase 1, and in meiosis 2, or in meiosis 2 especially in anaphase 2. Now here what happens? Centromere will be separated and sister chromatids also will be separated. Each containing now here each containing centromere, because splitting done, splitting of centromere is there. This is the centromere where splitting takes place in meiosis anaphase 2. Okay. Then what we get as a result? We got 4 germ cells. So now come on that. Germ cells. So these is having in linear position and become germ cell. So these are 4 germ cells. Okay. Now here 2 things we have to remember. Homologous chromosome separated at anaphase 1, but sister chromatids with centromere splitting or separated at anaphase 2. Okay. And finally, how many germ cells we got? That is 4 germ cells we got. Okay. Now next we will see this experimental verification. Okay, this experimental verification proved by many scientists. Among them, T.H. Morgan, Thomas Hunt Morgan, having the very much responsibility. Okay, they did many work on that, and that's why he is known as father of experimental genetics. Okay, so come on that experimental verification. So what we can say, experimental verification of this theory, chromosomal theory of inheritance, was given by first T.H. Morgan, then another scientist were there, Sturtevant, another scientist were there, Bridges. Okay, so we have to see experimental verification of chromosomal theory of inheritance. Was given by Thomas Hunt Morgan, and in short it is also telling T.H. Morgan. Another scientist, Sturtevant and Bridges. Okay, so these are the scientists who provide experimental verification on the basis of what he used that variation. So on the basis of variation occurs in sexual reproduction. So on the basis of variation occurs in sexual reproduction. Okay, that lead to variation in sexual reproduction. So this, but you have to remember that experimental verification given by mostly what? T.H. Morgan. Okay, now this Morgan chosen tiny fly, and that name is fruit fly, Drosophila melanogaster. Okay, what is that? Drosophila melanogaster. So Morgan chosen fruit fly, Drosophila melanogaster. This is the scientific name of fruit fly, Drosophila melanogaster. Okay, so Morgan chosen fruit fly for their experiment, over pea, over garden pea, chosen by Mendel. Okay, this scientist Morgan for their experiment chosen what? Fruit fly, and garden pea chosen by, this is garden pea, and garden pea chosen by what? Mendel. Now question is there, why this Morgan chosen only tiny fruit fly? Okay, why we are telling tiny? Tiny because their size is 2 mm. So this is very small size tiny flower. Okay, so whatever the reason is that, so we have to see that. So first reason is that they could be grown on simple, okay, they could be grown on simple synthetic medium. So first is fruit fly can be grown in simple synthetic medium in laboratory. Even in ripened banana. Okay, even in ripened banana or ripened mango. So first benefit is that simply they can be grown in laboratory. Okay. So next we have to see what are the advantage over P. Why this Morgan chosen only Drosophila? So first we seen that they can be grown in simple synthetic medium. Okay. Next is their life cycle is very small. Okay. So they complete their life cycle within 2 weeks. Okay, 15 days. So they complete their life cycle within 2 weeks. 14 days. Okay. So this tiny fruit fly life cycle can be completed within 2 weeks. Okay. And what another benefit? A single mating produces large number of progeny flies. So third is a single mating produces large number of fruit flies. Okay, a single mating produces large number of fruit flies so that experiment can be done easily. Okay, next is what? There is a clear determination, differentiation of sexes, male as well as female Drosophila. So what we can say? There was a clear differentiation between male Drosophila, male Drosophila and female Drosophila. Okay, that is having clear differentiation. The female Drosophila is larger and male Drosophila is smaller in size. Female Drosophila is larger in size compared to male Drosophila. Okay, so easily these fruit flies can be distinguished. Okay, then what is that? Many heredity variations occurs that can be seen with simple low microscope. So what we can see? Many genetic hereditary variations can be easily observed with the help of low microscope. Okay, with the help of low microscope or low power microscope, both we can say. So this can be easily observed. Okay, easily diagnosed. Okay, and what is that? this also having hermaphrodite condition